Hi, my name is Maya Cole. I'm a student at Arizona State University making this presentation on behalf of the BIO282 Lab course. I'll be comparing a media source to a primary source as shown on this slide. The topic in discussion is who gets sickest from COVID-19. We will be talking about the HLA gene and how it relates to COVID-19 severity. Throughout this article, I will be referencing Who Gets Sickest from COVID-19 by Elizabeth Svoboda and the association between HLA gene polymorphisms and mortality of COVID-19 by Yusuk Chamida et al. Starting off, I will first dive into the media article. Svoboda is trying to explain to her audience, the general public, potential causes for the differences in symptoms and severity among individuals diagnosed with COVID. At the top right, I have a quote that is directly from the article. The suspected cause for the disparity starts with the HLA complex. In simple terms, that means the cells that help fight infections as part of the immune system. The author also mentions trained immunity, where multiple exposures allow the body to have a better immune response. She claims lifestyle can also have an effect on the severity of COVID-19 from a receptor called ACE2, and that is more common in smokers and is the site of where the virus enters the body. Finally, she sums it up with the cytokine storm, which is another type of immune response that causes inflammation and is associated with low T-cell count levels. This is all good information, but is it valid? The author develops ethos by mentioning a scientist at the University of Hong Kong. However, she is very broad with her sources, using terms like experts say and scientists report. The information she provides is very broad and non-specific in terms of how these issues affect COVID symptoms. Lastly, the article was published in April of 2020, but the pandemic was declared in March of 2020, making her claims a little premature. Moving on to the research article, their hypothesis states that HLA genotype might impact the differences in morbidity and mortality of COVID-19 due to differences in the T-cell response. They test this hypothesis by utilizing a silico analysis and compiling public data records of allele frequency of HLA. They discovered individuals with the genotypes HLA-A1101 and HLA-A2402 do in fact have different antiviral responses to COVID in comparison to individuals with the HLA-A0202 genotype. As part of their research, they compiled frequencies of different HLA genotypes by country and chose to study the most frequent HLA alleles. This is just a continuation of the previous slide showing the frequencies. Feel free to pause and look over the data. Methods and analysis. The researchers compiled COVID-19 cases and death counts from the World Health Organization on two separate dates to ensure accuracy as time passed with the study. They utilized HLA peptide binding algorithms to calculate the number of SARS peptides bound by each allele. They also used this T-cell prediction tool to determine how many T-cells were present with each genotype. They did an epitope analysis of HLA to see the affinity of HLA to bind across the SARS proteome. An epitope is just the part of the antigen molecule in which the antibody attaches itself to, so they did an analysis of that for the HLA alleles. Lastly, they used a program stated at the bottom right to compare the deaths in different countries based on their genotype to compile their data and draw conclusions. Results. They found that countries with higher frequencies of HLA-A0201 genotypes accounted for 1,842 cases per million population, while countries with HLA-A2402 or 1101 genotypes only accounted for 97 cases per million people. So their hypothesis was mostly correct because the 0201 gene did account for more COVID cases, while the 2402 and 1101 genotypes did not. They followed up by doing a statistical analysis on the data and found that the p-value was 0.1, proving the data is statistically significant. They did the same thing for their other result, stating that HLA-A0201 accounts for 98 deaths compared to 2.5 deaths compared to all other genotypes. This had a p-value of 0.003, which is highly statistically significant. This table is just another way to organize the statistical analysis, giving a little more information on standard error and confidence intervals. This slide shows the results from the T-cell antigen analysis. The 0201 and 1101 genotypes have much lower T-cell antigens than the 2402 genotype. To tie everything together, we see that the media source contained borrowed information while the primary source has original material. The media source was much easier to understand but was oversimplified compared to the primary source and contains bias. Overall, the information was sort of correct, but I would lean towards the primary source for this type of research question.